Hi, I'm Matt Wimsatt. I'm the staff medical illustrator and animator at the Jackson Laboratory, which I'm sure you've all heard of. Actually, how many people have heard of the Jackson Laboratory? Oh, really? Okay. I was hired actually as part of my job to bring more attention to the lab. I do a number of things there. What I want to talk about today is volume image data, which a couple of the presentations just preceding mine have chatted about, and media development in a research setting at the laboratory. Jackson Laboratory was founded in 1929 by seed money from a bunch of auto moguls, actually, and then Clarence Cook Little founded the lab in Bar Harbor, where he had a summer lab from his Harvard days. The lab has about 60 plus researchers, principal investigators, scattered over three locations. I'm in Farmington in Connecticut on the UConn Health Campus, but the original lab is in Bar Harbor, which I go up to about once a quarter. It's right on the edge of Acadia National Park. It's quite nice. And then the other facilities out in Sacramento in California. The principal investigators that are working there, they utilize a number of different, as any principal investigators do, use a number of different methods in their research. Volume imaging is just one component. There's things split up between the wet lab and the dry lab, computational considerations, behavioral assays, patient-derived xenografts, which is a big part of what happens up at JAX. But if there is something that volume data can help elucidate with their research and their investigations, then it's something that's employed. I'm working with about 6 or 7 different labs up there right now with initial conversations, and I'm flirting with about 3 to get more. So, but before, actually, this has kind of been answered a bit because the last couple presentations have talked about volume data. Basile's work is related to the cochlear epithelial cells, and in particular the stereocilia, which are seen here in blue. It's, as a slide showed in an earlier presentation, serial slices through a number of different medium, electron microscopy, CT, can then be translated into volume data that people can utilize. As well, a number of the researchers up there are really good about splitting out the data onto different channels, and they do it really quite well. So Basile in particular is researching the stereocilia, so he can identify that very quickly with just the stains and the different mechanisms he's using. Did want to talk about Mount Sinai. For a number of years, I actually did work in New York before going up to the Jackson Laboratory, and I worked in particular with a couple different people here. John Morrison in particular, who's now out at UC Davis and runs the National Primate Research Institution out there. That neuron actually was developed here at Sinai, and I was involved I was the director, the volumetric production director at a medical creative group here in New York for a number of years. Actually, one of my former colleagues is here as well, Anne Canapari. And we did, while we were there working with volume data for different deliverables for different clients. This was a pyramidal neuron that John was working on here when he was actually a dean here at Mount Sinai with memory loss. And the actual image figure, which was in, I believe, is Nature Neuroscience, I forget the actual publication. But this was produced in a range of different programs by a small team of people. I was involved. I did actually some of the production, but then also was the liaison directly with John and with some of the people in his lab in putting that together. It took a long time to do. It was actually unstitched at the time, where most data now is stitched. So, but what it's showing, I'm sorry, is memory loss and aging. So on the left, you see a pyramidal neuron. I think it's a prefrontal cortex neuron in a rhesus monkey, showing the heavily studded dendritic spines on the left, whereas the aged individual on the right, you can see that the dendritic spines are quite reduced. So this was really an exciting project to work with him, and we were essentially working with him to help quantify and the data, but making sure not to introduce any bias, which was always an important thing, but serving as another way, another avenue, another aspect of the research to help prove what they were trying to research. So that was great. That was a lot of fun. And I missed working with a lot of the folks here and some of the other folks I worked at different institutions with when I was at the creative group. Then I had the opportunity to go up to the laboratory up at JAX, and they were doing a lot of similar work up there. And so I was really excited, and I got up there and I did the sort of things that you do. I talked to people. I would try to, tell them like, oh, if you give me your volume data, I can do things with it. And it was an interesting sell to some folks. Some folks got it early on. I started this project, which is the main project I'm going to talk about, with Simon John's lab, and in particular Krish Kuzatel. What you're seeing here is a variety of volume data of the Schlemm's canal. And the Schlemm's canal is something that they're researching because they believe it to have a very important impact in glaucoma in particular. The Schlemm's canal is, as you can see in green, it's a structure, a canal, that circles the eye and plays a role in the aqueous flow between the different chambers in the eye. They're not exactly certain how that aqueous flow works. So we're using a lot of the volume data that they gave me and working with them. They wanted me to do a proper cartoon, is how they described it, of what, and this is the image that I initially started with, what's the structure like? And so using probably a dozen different volume data sets, this was one of the main ones. This was it's an electron microscopy shot, but the canal is kind of flat and round. And if you think about what I'd shown earlier, the illustration I'm talking about right now probably would comprise about that much of the canal. So it's just one little section out of the canal. But they just needed to show how the canal is structured, all the different functions. I wanted to show the final, which I couldn't do because it still hasn't been published. They also insisted on the final illustration being bright green over my strong objections. But so this really tastefully colored version was not what we used. We actually used a really bright green one. So you always don't have all your battles won. We ended up using also the data sets for overall lab communication purposes. So this was from the Search magazine, which is an internal publication that goes to all the donors that provide funding for the lab. It's kind of eye candy. The next project I was involved with, I'm still kind of ongoing, was Carolina Paluca's lab and Jan Martinique, who's a research assistant with her. This is still ongoing. I just wanted to kind of get a taste for this one, but they do a lot of immunotherapy work and cancer vaccines, and so they're very interested to know how tumors are growing and how they're serviced by blood vessels and lymph vessels as well. This is just a simple animation out of Imaris, but we're using simple clips like this, inserting them into overall video productions that are being done by a small creative group that I'm a part of at the lab. Also used for the annual review, for the annual report, which is a pretty big deal with all the financial folks. The lab as well has, I should have mentioned this earlier, it's a nonprofit institution, research institution. It's really a beautiful place in that they do a lot of hands-on work and they breed mice for sale, as probably a number of folks in the room know. And because of that, there's a big financial aspect to the lab, but all of the money as a nonprofit institution is rolled right back into the research. So it's really quite amazing. So because of that, they need imaging for communication purposes. And so I was able to help folks in developing imagery for that. This is probably the project I'm most excited about right now. I have a couple things to show from this. I actually have a direct link on my desktop to this lab's imaging server, which is like insane. It's something we always dreamt about when I was at the Medical Creative Group in New York that we tried to get for here at Sinai. I was just able to get this set up about a month ago. What you're seeing here is some of the data sets that this group is working with. They also do glaucoma, as you can see from the chair title. They do glaucoma research, but Gareth's lab is very interesting in that they're doing neuroscience work, but they're trying to find direct links between the brain and the eye. So in particular, they're trying to see, and the project I'm involved with right now, which I might come back, maybe talk about it next year if I'm invited, is trying to find comparisons between the angiogenesis patterns that you see within the brain and within the retina, and any correlations that they're there, and potentially the ability for any diagnostic testing that can be developed and helping for Alzheimer's. I have a quick little animation clip. Shows you some of the data. It's actually quite thin. This is some of the work that I've been doing with them, and this is a surface extraction of those neurons that you saw taken out of, I think, ImageJ, though I'm trying to find a different production pipeline. If anyone is familiar with 3D Slicer, I'm really interested to talk to them after the talk. And then based on that, this is a preliminary one. I've been receiving more and more data, and I'm looking to build on this. And essentially, the pipeline I have in place is to start building polygonal models for Gareth, for him to start understanding the different structures he's working with. And I'm trying to turn the corner on this, where I'll be working with the labs more and more in a researching setting, as opposed to just taking their images and being able to put together a nice eye candy that a lot of my colleagues use for very important publications for the lab. But I'm trying to bring it to a different level. When I was at the Medical Creative Group, there was a whole team of us, so it's just me up there. So with time, hopefully, and getting some procedures in place, I'll be able to be more effective. It's difficult because I have to juggle a lot of things. As people, I know a lot of medical illustrators here that work in service desk settings at different institutions. You have to juggle and balance a lot of things. So I'm not doing review article image figures or manuscript image figures or grant proposal image figures, which I do a lot of. I can allocate time into developing the volume data program up there at the Jackson Laboratory a bit more. So in the interest of keeping things brief and getting us back on time, I think I'll stop there.